My name is Kadambot Siddiq. I'm from the University of Western Australia. My co-chair will introduce.
Yeah, I am Kuldeep Singh, Director, National Bureau of Plant Genetic Resources, New Delhi.
Thank you. And it is my great pleasure to see the 10 years of progress made by ICRISAT and Rajiv and his team in this area. And it's one of the most important labs in the world where the genomic information is applied in the practical breeding program. I think that makes the uniqueness of this institute, and not many places in the world have got that pipeline connection from the basic research into the applied aspect. So that makes a huge difference. So we have 4 speakers in this session, and it's about genome and germplasm diversity. I think the morning session clearly gave some ideas behind some of the new technology, but the genetic germplasm is the major source of any variation, and how the genome approach can be used to mine the genetic resources. With that, I would like to introduce the first speaker, Professor Suk Ha Lee from the Seoul National University. He's the Dean of the faculty and has been working in the legumes for a long time. Professor Lee.
Thank you, everybody. First of all, I'd like to express sincere thanks to Dr. Rajiv and his staff for inviting me to attend this meeting. Actually, I took part in this meeting several times. The meeting was great, and personally, this meeting was very helpful for me to plan the next experiment in my laboratory. Today, I'm going to give a talk about high-resolution genome sequence of mungbean using PacBio long-read assembly. About 3 years ago, we published the draft genome sequence of mungbean. More recently, we planned to improve those genome sequence of mungbean using some new technology. Let me introduce briefly about mungbean crops. You may know mungbean very well, but mungbean is some legume crop, mainly grown in India, China, Southeastern Asia, including Myanmar, Cambodia, Vietnam, Thailand, and Indonesia. And India is the first producer of the mungbean in the world. Second is China. And the mungbean is a good source of dietary protein and other nutrients. As you see here in these slides, mungbean can be used for producing mungbean sprout, mungbean noodle, and inside for dumpling and pancakes. In spite of importance of mungbean crops, the breeding program for the mungbean is less developed. Most of the mungbean breeding program, especially in Southeastern Asia, they are using the mungbean breeding was conducted by some pure line selection from local cultivars or introduction of mungbean genotypes from many other countries, rather than artificial hybridization. So very limited number of research institutes in Southeastern Asia can conduct some artificial cross-breeding program in mungbean. As I mentioned earlier, we sequenced the genome. We published genome of mungbean draft genome sequence. We selected the VC 1973A that is developed at the ABRDC in Taiwan. This genotype can be grown widely from low latitude Thailand to high latitude Canada. And we sequenced the 431 megabase. This is going to be about 80% of the estimated genome size. The M50 length of scaffold is about 16 megabase using using 11 high-seq sequence data. And genotyping by sequencing was used for constructing a genetic map with 190 lines in the populations. We constructed 11 linkage groups with about 1,300 Smp markers. And we were able to identify 22,427 high confidence genes in the mungbean genome. We plan to improve mungbean reference genome sequence using some PacBio-long assembly, as I mentioned earlier. And the same genotype, VC1973A, was selected, and the DNA was extracted. The DNA of VC1973A was sequenced by PacBio RS2 platforms. And the mapping population, we have 187 recombinant inbred lines were resequenced using PacBio. And this is derived from a cross between VC1973A and the Korean landrace V2934A4 collected near Seoul City. And all these lines were resequenced by Element HiSeq, 4,000 platforms. And let me introduce just briefly low-read statistics. And using PacBio sequencing system, about 7 million reads were produced. This is about 68 sequencing depths. And also, for 187 recombinant inbred lines were resequenced using Element HiSeq. Then this is each line has about 12 sequencing depths to construct a genetic map. And this combining of this PacBio long-read assembly and some genetic map, we were able to produce new reference genome assembly. And here, as you see here, about M50 of quantic scaffold length is about 1.6 megabase for the previous data, but it was much improved, 5.1 megabase. And some quantic, and the previous quantic revealed that we have 431 megabase was recovered in the quantic, but the long-read assembly and our newly genetic map can improve 473 megabase. And as you see here, the genetic map, we have by resequencing data, we were able to map about 9,000 Smp markers for the mungbean genome, the fully saturated map in these mungbean crops. And just briefly, the M50 length of the super scaffold is about 47 megabase. This is much improved by this study. And you can see some comparison of genome assembly of our previous reference genome and the new reference genome. As you see here, about 1,300 markers were mapped on this genetic map, and this is about 9,000 markers. And here, some of the fragments in the previous markers in the different chromosome length chromosomes were converged into one chromosomes, and also some fragment in one chromosome was diverged into two different chromosomes. So this was confirmed by the comparison of other soybean crops. So this newly assembled genome of the mungbean was much more improved by comparison of those other legume genome sequence data. And just in summary, the total base was improved over the context for the 431 megabase was recovered in the previous assembled, but now 473, and the M50 length was improved from 1.6 to 5.2 megabase, and the scaffold, the number of scaffolds is much reduced from 2,748 to 330, and the high resolution genetic map was mapped, and also we were able to find about 37,000 protein coding genes were identified in this new reference genome sequence data. And using this genome sequence data, we were trying to evaluate some genetic diversity of mungbean collections. About 276 accessions collected from 24 different countries. Out of 276 accessions is 233 cultivate mungbean and 42 wild mungbean, and we used one different Vigna species as an outgroup. So the mungbean cultivars have 44.6% nucleotide diversity of wild accessions. We know where soybean allied cultivars have 35% nucleotide diversity of wild accessions. So there is some rooms for improving some mungbean genotypes by selection of the desirable genes in the mungbean genotypes. And the population structures were examined by Bayesian inference using 1,000 random Smp markers. And the optimal, at the optimal k is equal to 3, 1 wild and 2 cultivar subpopulations can be grouped by these population structures. So we found very strong population structures between wild and sub-cultivar cultivar populations. As you see here in this slide, is depending on the geographic regions, there is significant difference in these dominant subpopulation genotypes in the mungbean collections. So we are also interested in the most differentiated, regions, genomic regions between cultivars and the wild accessions to know some genetics of domestications. And the 1.5% of the genome with about 470 genes showed a highly differentiation between wild and cultivars. However, we do not know exactly this gene is the still key regions, key genes in the selected genomic regions for domestication. So we are now trying to see what the gene is in their genomic regions in detail. Now we are also using this genotyping by sequencing data. Also some phenotypic data were collected in the fields. We are trying to GWAS some association analysis of genotyping data with some phenotypic data. So phenotypic data were collected widely, but we are very interested in the synchronous pod production. So I'm going to put a little bit emphasis on this synchronous pod maturity production. Let me introduce briefly about the synchronous pod maturity in mungbean. And most of the mungbean genotype is characterized with non-synchronous pod maturity. So as you see here, some pod is already mature, but now you can see some young pods also. In here, looks like a synchronous pod maturity, but here you have some new flowers. They produce some another pods. So because of the non-synchronous maturity, so the mungbean require higher labor cost and a low harvest index. So farmers should harvest mungbean 2 or 3 times during the harvest seasons. So many farmers don't like to grow mungbean because of this non-synchronous maturity. So we were trying to harvest every week some mature produce for all the collections to know some synchronous pod maturity. This is the, we developed some parameter for quantifying synchrony. This is the number of pods matured each week. So this is two different genotypes. One, if you harvest one pods, then you can see another peak in the later. So probably 7 weeks later after the first harvest. So because of two harvests, then farmers may should harvest 2 times. So this genotype is non-synchronous maturity. However, in this genotype, you can, if you harvest the second week of the second week, then the other, you cannot see the developed more, you need not harvest anymore. This is the some accumulated pod numbers, the bar representing the accumulated pod numbers. So the GWAS results for this this is the two GWAS methods, GAPIT and test were used for to find this some gene QTR. And this is the C17 total. We found only one markers only on loci. We found only on loci involved in the some non-synchronous pod maturity. In addition to this GWAS, we were also trying to identify QTR using some linkage group we constructed. And here you can see 9,000 markers are mapped on this linkage group in one populations. And this also one Sonan, the VC19738 developed at ABRDC showed a high synchronous maturity. You can harvest just only 1 times at the 2 weeks after harvest, then not any more mature pods. However, the other variety pattern lines has 2 peaks, so you have to harvest at least 2 or 3 times for these genotypes. And we checked all the lines, progeny lines, by harvesting some pods every week 8 times. Then we used that data to estimate parameters. But this experiment was conducted in different times in different persons. We used the different parameters to see the synchronous maturity of pods. This Smp Index was widely used in mungbean breeders in Southeastern Asia, and they used this parameter. The yield of the peak week was added with a higher yield between flanking weeks of the peak week, and divided by total yield. So this is the way. This is the YH, and this is the YHF. This peak yield, this yield was divided by total yield. So this is called the Smp Index. So you can see some histogram of synchronous pod maturity. The VC19738 was near to 1, representing the synchronous maturity. V2984 has the non-synchronous maturity. So using this phenotypic data, we identified some QTR for SNPs, and we found 2 markers, 2 genomic regions which is involved in pod maturity. I'm sorry to inform you that direct comparison of the QTR locations can be made between GWAS and the linkage group result, because we use the different parameters, and because of the limited number of samples, we cannot have the same type of data collected. So, but anyway, the identified QTRs, some QTRs in these linkage group studies, And also, some GWAS can be used for improving the pod maturity, synchronous pod maturity in mungbean. Now we launched some breeding program based on the genetic diversity and some yield in Southeastern Asia. We crossed the mungbean, the artificial cross were made in our university, and the advanced generation in our university farm, and some advanced generation was sent to the Cambodia and the Vietnam, and the advanced generation also tested and selected for desirable genotypes of the mungbean. We launched this program about 3 years ago, and some elite lines were sent again to the Vietnam to test some adaptation of the mungbean varieties we breed. So this is, but the problem here, you can grow the mungbean 3 times a year because no winter, so you can advance generation very easily in Cambodia and Vietnam. However, there is some seasonal variation. So if you, depending on the planting times, we are in trouble to find some desirable best genotypes in because of the seasonal variations. But anyway, we found some elite lines, and the lines were now will be grown in Vietnam a little bit massively. So I'd like to finish my talk by actually Dani was working for the GWAS from Indonesia, and Han Wei-jung was working for the genome assembly and some linkage group study. And Eunsu, she's here, she's working for some synchronous pod maturity. And I'd like to finish my talk. Thank you.
Thank you, Professor Lee. Well done. You have served quite a bit of time there. Questions, please, very brief, and also mention your name and the institution where you're coming from, please.
Myself, Dr. Chiralu, from Professor Deshankar, Telangana State Agriculture University, Telangana, India. Very good information has been provided, though it is a small crop, grown everywhere with the utmost least interest by many of the farmers. Even then, throughout the country in India, in one or the other season, the mungbean is grown. Because of that, the soil fertility and carbon content in the soil is being maintained. So my question is that, as you rightly said, regarding the climate change, and one of the criteria that you are looking into it, but in rainy season mostly, in summer and also in the winter season, this mungbean is grown. But in rainy season, mungbean is generally caught by the rains. And almost more than 70% of the yield losses will be there. And as a result, farmers are not realizing good economic results. Do you have, just like synchronous maturity, do you have any information regarding the pre-harvest sprouting or weather damage in mungbean with reference to genomic studies is concerned? Thank you.
So the question is about pre-harvest sprouting, so because of the monsoon rains.
So actually, just we start this mungbean breeding program very soon. As you know, I mentioned about seasonal variation of the mungbean crops, but depending on the weather conditions, there is some problem in pre-sprouting of the mungbean. However, so in this case, we just we used the Cambodia and Vietnam to advance generations. We did not have enough time to see the pre-sprout. So pre-sprout has a big problem because the quality of the seed is lowered. And also, we just see the synchronous pod maturity, but after this breeding program, we figured out some kind of seed coat color or seed coat some polishiness or any other important agronomic trait should be emphasized in the future. But we just started this breeding program.
Thank you. Any other questions? Yeah, there is 2 questions. Debrief the questions, please.
I'm Dr. Sanjeev Gupta. I'm coordinating Indian program on mungbean. Mungbean is basically a short day plant and very sensitive to photoperiods and thermoperiods. So since in many countries it is growing over seasons, I think this can be a good trait to be targeted photothermo insensitivity in this crop.
Actually, I mentioned the VC 1973A. So you can grow VC 1973A from low latitude Thailand to high latitude Canada. However, I don't think it's I don't think it is photosensitive, insensitive variety. Depending on the planting time, it should be they should have a different agronomic trait. But in my opinion, the mungbean is not much more photosensitive to soybean. For example, soybean, the high latitude soybean, if you grow the high latitude soybean to the low latitude, you cannot, you just within plant height, small plant height can flower. So I think mungbean is still, compared with the soybean crops, mungbean is a little bit photo-insensitive than soybean plant. So still, you can grow a little bit widely than mungbean than soybean plant.
Thank you. There's one question here.
Hi, I am Aditya, the mungbean breeder at Indian Institute of Pulse Research, Kanpur. When we talk of nucleotide diversity in mungbean in comparison to wild accessions, and we say that it is 45-44%. So do you mean that it is in relation to Vigna glabrescens only, or you have taken into consideration other related wild species just like Vigna sylvestris?
So actually, depending on the collections and or depending on the genotypes, the nucleotide diversity may be varied. However, that just because the soybean, many breeders put a lot of effort to improve the soybean crops. So just by comparing of the mungbean, but when in comparison with the soybean varieties, mungbean has a little more higher nucleotide diversity because breeding effort can be done to improve some mungbean to improve. So that's my just basic thinking for the mungbean.
Thank you, Professor Lee. Please join me in thanking Professor Lee.